Okay, biochemists, strap in because we're going to go over the steps of glycolysis in our video today. So glycolysis is the process of breaking down glucose, and the end game here is we're getting towards pyruvate. So pyruvate is the end product of all of this, which is going to help us to go into the citric acid cycle because the pyruvate can be formed into the acetyl-CoA, and then the acetyl-CoA is what starts the citric acid cycle. So in order to get there, we have to start with glucose. And so we're going to go step by step and talk about the different reactants, products, and kind of how we get there, because it looks a little bit complicated, but all it is this breaking down, this catabolism of larger molecules into component pieces, doing some rearrangement, and then ending up ultimately with pyruvate. That's kind of the name of the game. Okay. So the first step is phosphorylation. As the name implies, that means that we are getting a phosphate, we are phosphorylating, we are adding a phosphate to something. That something is glucose. So glucose is hanging around in my bloodstream. So I ate something, my saliva broke down my starches into smaller pieces, and those glucose molecules are now going from my bloodstream. They get sucked into cells, and as soon as they do, they get phosphorylated. And as soon as they get phosphorylated, then they're stuck in the cell, and then all of the magic can happen from there. So the place that they get phosphorylated, though, is from ATP. So I have ATP that comes in and goes from ATP to ADP. So that's where that first phosphate comes from, is from my ATP. And we'll try to keep these kind of color-coded as we go. Okay, so it cost me an ATP to phosphorylate this glucose, and so now I have this phosphate group sticking off the end, and now I have what is called glucose 6-phosphate. So it's still glucose 6 because of where the phosphate is located. Okay, so the glucose molecule is unchanged, but on that 6th carbon there, that is the one that's poking off the top, has a phosphate group. All right, so that's step 1. Now we're stuck in the cell. Now that glucose 6-phosphate is my reactant here. Now it's going to undergo some isomerization and become a 5-membered ring instead of a 6-membered ring. So glucose is 6-membered. It's a hexose. It's an aldohexose because it's an aldehyde, and it becomes a fructose ring, which is a keto is instead of the aldo. So that's kind of a difference in this thing. So we still have the same number of carbons though, so it's still a hexose, right? We just have them in different orientations, a different isomer of it, because now I have a 5-membered ring instead of a 6-membered ring. So now the functional difference between the two is, instead of just having one group sticking off of one side, now I have them sticking off of both sides. Okay, so this is phosphated, so this is fructose 6-phosphate. Okay, so now we just underdid some isomerization. Of course, this is enzyme catalyzed, so it would be an isomerase that does this. And now we have phosphorylation again. So if we look at my fructose here, here's my fructose 6-phosphate. Now I have a phosphate that can stick onto this group right here, right? So that hydrogen at the end pops off, and we end up with this phosphate. That's this phosphorylation process. Well, where does that come from? That comes from another ATP. So my ATP has to come in, and it costs me an ATP to give me my ADP. And again, that phosphate group that sticks on the other side, so I already have this one accounted for that came from the first ATP in the first step. Now this one is going to account for this guy in the second step, or in the third step after isomerization, after it does some wiggling around. Okay, so now I have fructose 6-phosphate going to fructose 1,6-bisphosphate, fructose 1,6, bis for 2, phosphate. Okay, so on the 1 and the 6, so my 1 carbon is down here, the one that's sticking off here, my 6 carbon's up here. So fructose 1,6-bisphosphate. Okay, so now, so far, this whole process, these first 3 steps of 10, have cost me 2 ATP already. So in these first actually 5 steps, we call this whole part of it the energy investment phase. So we're investing energy in steps 1 and step 3 by using up these ATP in these phosphorylation steps. Okay, so 1 and 3 are my energy investment phases or steps in this overall process. And they kind of lump them all together and say, well, the first half of glycolysis, the first 5 steps are energy investing, and the second 5 steps are energy generating, meaning that we're forming ATP instead of using it up. But we've already used up our 2 ATP, and that's all we're going to be using in this process. So for every 1 glucose, I have to use up 2 ATP in the glycolysis process. Okay, now here's my fructose 1,6-bisphosphate, and now I have a step of cleavage. And so there's an aldolase that cleaves this thing, and it cleaves between carbons 3 and 4. So that would be 1, 2, 3, and 4. So it's going to cut right here. And then when this thing cuts, this cleavage step, then it's going to cut it into 2 different pieces. And so there's these 2 pieces here. This top one is called dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Such a mouthful, these biochemical names. And then the bottom one here is D, and this is going to be confusing, but I'll tell you about it as soon as I get the whole name out here. D, glyceraldehyde, 3-phosphate. And of course these would all be on the same line if I had enough room, but alas. Okay, so these are my 2 products that I get when I split this thing into 2 component pieces. So dihydroxyacetone and then phosphate, okay. And then this guy, so we're kind of looking at commonalities, looking at how they're different. We split this thing in 2 parts so we can kind of clarify where they came from. So one of the phosphate groups ended up on the end here. The other one ended up on the end here. Okay, we can kind of map out how that worked out. Okay, now you might be saying, but Ms. Gross, this is the D isomer, which means that my hydroxyl group should be sticking off to the right. But you'll note that my aldehyde group here at the end is sticking out to the left. So by convention, it's the same side as this one. So it is the D. It's just in the way that I wrote it. So kind of picture that flipped over. And that's my D-glyceraldehyde 3-phosphate. So you would be correct that yes, that's confusing, but that's the way that I copied it out of the textbook. So you can blame someone else. Okay, so that's step 4, that's cleavage. And now we have isomerization between those 2. So now I have those 2 products, I can go back and forth between them. And preferentially, this thing kind of likes to shift towards this general direction. So we like this D-glyceraldehyde 3-phosphate. This is kind of the one that's preferred. And this is the one that gets used in step 6. So the thing is that as this thing is used up in step 6, then Le Chatelier's principle says, because it's an isomerization reaction, so there's an isomerase, we can flip back and forth between these things, there's an equilibrium. Le Chatelier says if I remove some of this product, so some of this is getting used up in step 6, then I'm taking some of this away, that shifts the equilibrium towards reactants, and functionally all of it is going to ultimately be this D-glyceraldehyde 3-phosphate. That's sort of the magic product here. And even if there is some of this, then it's going to keep being pushed in this direction from an equilibrium standpoint as we pull this one out and get used up. Because this is the one that gets used up, not this dihydroxyacetone phosphate. Clear as mud? Yes. Okay. So those are my first 5 steps. So the first 5 steps are my energy investment steps, and now we get into kind of where the magic happens. Well, it's all magic, right? It's science. So step 6 is oxidation and phosphorylation. We end up with my oxidizing agent here, meaning the thing that's getting reduced, right? That's that thing about redox reactions, right? So I have my NAD+, and I have my other group here that's going to donate my phosphate. So that's my donating group. And then you'll see that I kind of wrote this funky, again, I ran out of room on the side of my page. So it's the same molecule that I had to start off with. that D-glyceraldehyde 3-phosphate. Now my NAD plus and my HOPO3 2- are going to come in, and then I'm going to get some reduction that happens here from my oxidizing agent, and now this is being reduced or being oxidized. This guy's being reduced, and then we end up with this phosphate group here on the end. So then what we have left over is my reduced NAD plus to NADH and the hydrogen ion that's left over. Okay, so if we're kind of looking at what happened here, one of these hydrogens is going to mess with this guy, one of these hydrogens will be left over, everything kind of balances out. So what we ended up with is we had an oxidation step, so the oxidation of the molecule, reduction of my coenzyme here, and phosphorylation in the addition of the phosphate group at the end. Now I've got 2. So since I have 2, this is now called 1,3-bis for 2-phosphoglycerate. Such a mouthful. And then my NADH and my H+. Now that thing's going to be phosphorylated here, so we're going to transfer that phosphate group. And where am I going to transfer it? Well, it looks like in this step I'm going from 2 phosphate groups to just 1, so I'm losing this one. Well, where does it go? It goes on an ADP, which then forms ATP. Here's the generation part. Woohoo, I made some ATP. Okay, now in step 8, we have some isomerization here. So the product that formed in that last one was the 3-phosphoglycerate. So 3-phosphoglycerate. And now isomerization, again, we're just rearranging the thing. So this is going to be an isomerase that does this. So we're going to do kind of a swappage here between these guys. That puts my phosphate group up here, my hydroxyl group down here. Now instead of being on the 3rd carbon, so again we're counting away from this carboxylic acid here, so 1, 2, 3. So 1, 2, 2-phosphoglycerate. Okay. Now the reason we do that is because now we're positioned to have this hydroxyl group at the end. This hydroxyl group at the end is more reactive. It's more able to do some chemistry. So in step 9, that hydroxyl group at the end and this hydrogen are going to leave in a dehydration reaction. So we get some water that leaves. And what I have left is called phosphoenol pyruvate. Phosphoenol pyruvate. So it's a pyruvate with a phosphoenol on it. And this is actually enzyme catalyzed by enolase. Okay. Step 9, so we just got rid of that water, and now we have this kind of precursor to pyruvate. So in step 10, we're going to do a phosphate transfer. Well, where does the phosphate go? I had the phosphate on my phosphoenol pyruvate, and now all I have left is pyruvate. So what happens? Well, the same thing that happened before. An ADP is going to come along and then pick up that phosphate group, forming ATP. And again, woohoo, I made some ATP. All right, now the net result of all of this is going to be a net 2 ATP, because this is happening twice at each of those steps along the way. So while it took 2 to make those first 5 steps happen, I get 4 ATP in the second 5 steps of the process. And so we have a net 2 ATP at the end of it, as well as the pyruvate, which then can go into make our acetyl-CoA, which then goes into the citric acid cycle. Okay, and then we get more ATP from that process as well, but this is just the first step in metabolism, breaking down 1 glucose. So 1 glucose, we consume 2 ATP, and we get 2 ATP net out of it. 2 and then 4, net of 2. Okay. So that was a lot. I'm going to need some, speaking of glucose, I'm going to need some sugar after all that. If you have any questions on this, don't hesitate to reach out. Otherwise, I'll talk to you again soon.